And now we move to the last part of our workshop, and I'm very glad that I can welcome here my colleagues Eva Chocholova, Eliška Svobodová, who are the authors of our new exposition on genetics. So I suppose that the last part will be also practical and interactive, because we would like to show you our new exposition and to show you how we work here, how we present our programs. So Eva and Eliška, microphone is yours.
I have way too many mics. Oh, third one. Does this? Yeah, perfect. Okay, perfect. Hi, so my name is Eva Chocholova, and I am a PhD student at Masarykova University. What's happening? Wait, sorry. Okay. It has its own life right now. Before I start our presentation on sharing the fascination with genetics in Mendel Museum, I would like to say a bit about our motivation, because life of Mendel is very inspiring, as we all know after celebrating it for the whole year. But I would like to share a point of inspiration that I personally take when talking about science, when sharing my passion and my fascination with how the world works. And that's the teachers Mendel had. And that's who you might be or will be, or you can empower other people to be. Because if we didn't have all the amazing teachers that Mendel had, we probably wouldn't have Mendel, the father of genetics, but Mendel, the farmer, and we would not know about him. So we can be part of making a genius, even if we are not the geniuses ourselves. We can kindle the spark and the fascination, the curiosity in children, or even in other teachers that can afterwards help children with starting science. And even if no one ever hears our name in the future, we did something that was meaningful, and that's why we want to share our fascination with science, why we did the exhibition. So our museum. So we will talk about basically two parts. Me and Eliška will talk about the new exhibition that was opened just this May, that is called Genetics, a Fascinating Journey into the Cell Nucleus. We decided to build this exhibition to give more space to not only what Mendel did, because we in the older part of the exhibition that is about Mendel's life and his work, there was not enough space to explain the laws. And I think it is important, especially since we're connected to university, to tell the scientific story, not only the life story, which is very inspiring in itself, as I said. And we wanted to put the Mendelian genetics into context. As our colleagues previously said, we wanted to show what had to be done before, what had to be, or what happened afterwards, and what is Mendel's role in today's life and in today's science. How far did we get? And what is very connected to the exhibition are our programs for schools. Some of the activities we have in the exhibition are derived or somehow inspired by previous programs that we prepared in the museum, and vice versa. We're preparing new programs for schools that are based on the activities we have in the exhibition. So my colleague Maciej will talk more about the programs for schools, And in the afternoon, after lunch, there will be like an hour where you can take a tour through the museum yourself. You will already know what to look at, what you might be the most interested in. And we will have 3 spots where you can talk to us, discuss the activities a bit deeper, share your feedback, inspiration for future work, whatever comes. So this is the beginning of our exhibition. We wanted to give Mendel a bit more context. We wanted to show where genetics are happening. So that's why it's called Journey into the Cell Nucleus, because that's where things are happening in the eukaryotic cells, of course. So we start with entering the cell nucleus. We have multiple target groups. Of course, these are all school children or like university students and so on. The museum has plenty of activities for anyone who comes, even with like smaller children and so on. But we know that the teachers are very interested in how they can bring more interactivity into their genetics curriculum, basically, or like biology in general. It's very nice when you can take your students somewhere for the morning and explain whatever you explained during the lessons, explain it again with maybe slightly different view that we had as the authors, that might be slightly different from what the textbooks say, or like the way we explain it. We're not like presenting any alternative versions of biology. So the first group are primary schools that are mostly. We work with those mostly in the school programs because genetics is way too hard for them yet. My colleague will talk more about that probably, but we have one program that is about Mendel and his beekeeping, and second one that is about cells and that there is some inheritance. That's the basic idea they're taking out of it, I hope, that there is inheritance, that they look somehow like their parents, they can look for those traits and so on. Then the lower secondary schools, there are like the critical 11 through 15 years old. We just started a new program for them that is a bit more edgy maybe, and that can help spark interest in topics that might be maybe a bit too boring for them. So for them, as well as the secondary schools, we have a program about GMO that you'll see more later. And our 2 main target groups are secondary schools and university students. Actually, our university even has one genetics course that all of us went through, where we have to, as part of a lesson about Mendel and Mendelian genetics, you have to take a tour in this museum during the semester. So all the biology students at our university, all of them, come here. So we wanted to help them reinforce some of what they've learned, and it's an amazing opportunity to share this with them. And as the topic is quite more challenging, genetics, so secondary schools and universities are our primary target groups, I would say. So some people won't understand all the activities, and we think it's okay because we have something for them as well. So the chapters, as we call it in like the division in our exhibition, we have one room that is slightly, I don't want to say off topic, but a bit, is a Nobel Prize room that you will see, where we have a database of Nobel Prize laureates, just in case you don't find your favorite one. that happens every time. We only included those that were awarded the Nobel Prize for something that was built on Mendel's, I don't want to say on Mendel's laws, but on genetics in general, that went in Mendel's footsteps. So we don't even have all the biologists, just a disclaimer. Yeah, so we have a book where we have their signatures because they come sometimes for Mendel lectures that are held here in the museum or here in this refectory. Then we have 3 topics that are slightly more, I would say more modern, but like the basics of what cells are, we've known for quite a bit, but it's not like straight Mendelian genetics. So we wanted to show where the DNA is, where all of that is happening. So we talk a bit about cells. Then we have the main part that is Mendel and what we call no Mendel, which is basically non-Mendelian inheritance, just showing them that, okay, we do have these laws, we can still see them in some of our traits, but there's so much more in genetics that we've learned after Mendel. So we want to show them the exceptions as well, and show them some interesting examples. Then we have what basically came out of genetics. So the basics of molecular biology, we show some of the like basically school curriculum things like translation, transcription, and so on. And then we have a tiny small laboratory that is dedicated to show some of the equipment or some of the methods that we might encounter in a laboratory setting, such as electrophoresis and so on. For the form that we decided, of course, we have plenty of texts that anyone interested can read through, but what we really wanted to focus on was some physical activities, something that that you can touch, that you can play with, to bring more senses into learning, because we're surrounded with screens, and especially like we were preparing this during COVID, and everyone was sick of screens already. So we wanted to apply as many things in the physical form as we could, but of course the exhibition is a quite narrow and small space, so we do have multiple screens with apps for activities that would be way too complicated to make physically, or it would take so much space, or for like other reasons. So we have, I think, 6 screens with like 9 apps on them. And the content that I kind of covered some of that, but in the content we decided to focus on what is needed. So for the schools that come to revise, some of the principles are very basic, and you find them in all the like boring school books. But we wanted to show that as well, so that the teachers can again use it for revising the topics. So we have some of the topics that are needed for the school curriculums to go through. Then we have some that are just interesting, so that even a visitor who never encountered genetics can learn something new and can be interested in it. So we were asked by some visitors to add more about human genetics, because they were bored of looking at flies and things, because previously we tried to show examples that would not be, let's say it like, I don't want to say harmful, but that no one would take offense, maybe, because of human genetics. You know, you don't want someone coming and finding out they're not their parents' child, or you know, like there's lots of complicated things around that. But the visitors explicitly asked us that it would be amazing to see something that they can observe on themselves. So we actually have a self-observing station that my colleague will talk about. And then we want to show what is new. Such is the topic of GMO, for example, that we present, and many more. So this is the part where I ask Eliška to show you. What is it doing? Yeah, Eliška will tell you more.
So hello everyone, my name is Eliška Svobodová, and the same as my colleague, I'm a PhD student in genetics at Faculty of Science at Masarykova University. And as my colleague told you about our motivations and some ideas behind the exposition, my task was to show you some of the activities in more details and on which age group we aimed and what they can do with them. So we will start at the beginning of the exposition. You already heard that our exposition is starting by walking through into the cell nucleus. So probably you should learn something about cells. And for this we have an activity called Cell City. And I would say that this is one of the activities which is like a popularization of science in its finest. So we have like 2 pedestals, and you can see that on each of them there is a puzzle of a simplified cell, and next to it is a matching puzzle of a little city. And the main idea is to help children and scholars to understand and to help to memorize the functions of main organelles in cells by comparing them to different buildings in a city. So, for example, you have mitochondria, which are organelles producing the energy. So they are compared to little power plants in a city. Or the nucleus, which is like a cell headquarter, is compared to a city hall. Every organelle and its matching building has same color for some easier orientation. This activity can be easily used for younger visitors because it's basically a puzzle. But for high schoolers or some older and determined visitors, each of the pieces have a little text about what's the organelle about, what's the function of the building, so they can learn. And we have it in a programs for the schoolers. Another activity which can be used for quite a large variety of different age groups is a board where you can assemble your karyotype. Karyotype is a set of all chromosomes in cells, and actually this is a human karyotype. And for some younger visitors, it can again serve as a puzzle because each chromosomal pair has a different color or a different size, so they can easily like put them together or put the karyotype together, and they can even choose between the woman's or men's karyotype using the right chromosomes, and maybe showing them that the Y chromosome is actually in a shape of X and not in a shape of Y. And for the older visitors or high schoolers or college students, this board can also serve as something they can learn about some of the most famous chromosomal abnormalities. So they can like create a standard karyotype, and then by adding or removing some chromosomes, they can like mimic the events in people with, for example, Down syndrome, which is caused by an extra copy of chromosome 21. This activity, chromosomes in other organisms, is mainly focused on our smaller or younger visitors and maybe the general public, because we choose a number of usual as well as exotic or some kind of interesting, for example, the kangaroo, organisms like animals or plants, and we provided also the number of their chromosomes, which is sometimes quite far from the number we have as humans, and they can easily like rotate the carousel and see all of the organisms we have in there with their pictures. Of course, we are Mendel's museum, so we focused part of our exhibition to Mendel's work and research, which was so important for the development of genetics as a science. And we know that we already heard that the Mendel's laws are quite complicated to explain them, but we still wanted for everyone to learn something about it. So we created a set of different activities, so even smaller or younger visitors can learn something from them. I would say that the easiest one is something called Mendel's desk, where you have a paper pea flower and magnifying glass. And your task is to cut the stamens with pollen and then put with a brush a pollen from a different flower, which is actually a colorful ink, on the flower. And because this is something what Mendel was doing for quite a long time with significantly smaller flowers, but it was something we wanted the children would be able to do. Then we have a board with all 7 traits of genetic traits of peas that Mendel was observing, because I would say that the color and the shape of the peas are the most famous ones, but there were 7 of them. And as my colleague said, our college students have like mandatory to visit this museum, so we wanted for them to like see all of the traits. And then, of course, for mostly the college students, we have the explanation of the Mendel's law with some visual aid, or for example activity with the rails, where they can choose 1 allele for 2 genes from each parent, and they then can discuss how the specific genotype they choose will affect the flower, how the flower is going to look like. And as my colleague said, we also have some apps. So in this app, you can try the Mendel's experiment, which took months, but you can take it in a few minutes. And it's supposed to show the whole process of his experiments, from sorting the peas, planting the plant, and then taking care of it, because he was a meteorologist. So you have a different weather, and you should decide whether you will water the plant or not. And at the end, you will have to harvest and sort the peas. The game is fun for some younger visitors, but at the end of the app, there is a genetic explanation of how it works, about the genetics behind it. So it's even educational. Yeah, from peas we will move to another organisms, and most specifically humans. So we have a self-observing station, which is basically a mirror, but even with such a simple tool as a mirror, the visitors can observe some basic genetic traits, like the shape of their ear, the ability to roll their tongue, or something like that. And we have even some worksheets where they can note their observations about themselves, their friends, their families. Another example or activity is about blood groups. We have a wooden model of a red blood cell and 2 different kinds of surface antigens. You can see that they are different in color and in shape, and by choosing them or even combining them, they can create a different types of blood groups. There is also a text with some tables which explains the genetics behind it, but for some younger visitors it can also again work as a puzzle. And here's a tip for lazy visitors, because if you don't put any of the receptors into this, it's also a correct solution, because it's like zero blood group. In molecular biology, it's a little bit more complicated. So in this part, we mainly focused on college students and high schoolers, where they can learn about the flow of genetic information from DNA to RNA to protein. We even have a video which can be used as some visual aid for teachers, which can explain the process and then showing them pictures on the video. You can even do it yourself, and we have a set of mini games for this process. And this specific activity then serve as an example that they can try to translate the information written in white into the sequence of the protein using the genetic code, which is like the real one. Little Easter egg, the sequence is the actual partial sequence of molecule of hemoglobin. So they are translating something that is really like in our bodies. And even though anyone can try it, the deciphering the genetic code can be a little bit tricky. So that's why I said it's more likely for the older visitors. But actually, this is like a daily bread for geneticists. For example, just yesterday I was deciphering some patient sequences. So maybe it's a good way to try and to learn. And meanwhile, the adults and some older visitors are occupied by translating the proteins. We also have a translation for children. I would say this is like a hybrid of your activity that you were presenting with the monsters and activity that was presented by Hanka, because it's like a special code for every letter in a Czech alphabet, and then we have 3 different ciphers, and after they decipher them, they got the instruction how the monster should look like, and then they can like draw it, and we have it in an exhibition, so everyone can try it, and they can like use their creativity because they just get some simple instructions. Another very popular topic is GMO, sorry. So we have also a set of activities for this. This activity is more popular among younger visitors because you can try to connect nowadays crops like bananas or potatoes, which you can buy in supermarkets, with their ancestors, how they looked like 10,000 years ago, and with all those pictures. And then we have an app for this, where your task is to read some information about organism with some specific features, and you should decide whether this organism was created by using natural processes of breeding, or if it was made by in laboratory using the modern technology, so it's a GMO, or if it's just a nonsense we made just for fun. I would really love to have the slimming tea, but actually that's the nonsense one. And you can play the mini game. You get random 12, I think, 12 different organisms, and you can try to decide. Actually, those 2 activities will be prepared for you if you would like to see them in the afternoon, and me and my colleague will be there and be happy to assist you with any questions and anything. Last but not least, this is our newest app. It's a crime scene, and it's supposed to show you how important is genetics in a forensic science. You will meet there our forensic analyticist, Hannah. And you can choose from 3 different scenarios, and using the blood groups, or making the pedigrees, or analyzing the STR markers, you can help her to solve the crimes or solve the mysteries in a playful way. So this is again one of our apps, and that would be all from me.
Yeah, thank you. I think we will continue with our school programs, and we'll leave the questions for afterwards. Yeah. We have some questions prepared for you, actually, at the end of this. So just be ready.
Yeah, thank you. Hello, everyone. I'm glad to speak here. At first, I really like to thank you from Vienna that you enlightened me into the work I will present you. I'm kind of humbled by your presentations because our programs are really new. We are just 2 years into our developing and not much have been done, but we are trying to improve. I'm Matěj Přikryl and I master studies in molecular biology, and also I'm lecturer here, and I will talk about our programs. And do we have a presenter or not? Yeah, I'm sorry. So, as yeah, this won't work. Yeah, it works. Yeah, as Eva said before, this part of this workshop should be actually a workshop. So if you want, you can scan the QR code and go to Slido. And if you have any question, or maybe later in the presentation, I will say when's the time to open the Slido and put the questions here, or your opinions. It could be anything. And I'd like to ask if it's working, or yeah, great. That's perfect. So currently in the Mendel's Museum we have 3 programs. 2 of them are from primary school, and the third one about GMO is for secondary schools or middle schools, and that's the one I will be talking about today mostly, but I will mention the other 2 later. In the name, I put GMO in the comparison with breeding, and I will explain it later, because it's important for me to connect Mendel with GMO with one middleman, and that's breeding. Because you see, Mendel grew up on a farm and he planted some plants with his father, and he was beekeeping as a child, and everyday practice of every farmer in the 19th century was to somehow breed their crops or breed their animals. So Mendel himself was some kind of breeder, and then he came to Brno, and there was a center of breeding. From, I think, 18th century, there was a club of breeders. They were trying to breed fruit trees and etc. So he started his work in a place with breeding history. So Mendel was connected to breeding, and actually he tried to breed bees, but that's another story I will tell you later, maybe. And the connection between breeding and GMO is more functional, or the reason I'm connecting these 2 things is that probably the domestication and breeding itself is a reason why we are able to live on this planet with population over 7 billion, because without domesticated and bred species, we weren't here. We were not able to feed all the people on this planet, especially in Europe or USA. So that was in the past, and there is a future. So how will be able in the year 20 and 50, how we'll be able to feed 8 or 10 billion people on this planet, because breeding is not anymore sufficient. It's slow and expensive, and we have to move on and take some action. So maybe the solution is GMO, and that's the reason why we are talking in Mendel's museum about GMO. There is a connection with Mendel, but I think the biggest reason is to explain to the children that we have to talk about controversial themes because they're important for our lives. So, what aims of this program are, this is a list of things I want the students to take home as a take-home message. And the first one is that we can modify organisms in many ways. It's maybe unimaginable, but we can do pretty much everything we want to with our organisms, and there is a later question if it's stable, or if it will work, or if it's not dangerous. So that's the questions we're talking about also. Yeah, currently there is a number of approved GMO among normal products in our stores. There are GMO products, and we have to know that the GMO is around us already, and the students will take home the message that there are approved GMOs, and they do not need to fear the approved products because the process is like 10 years long, and the products we can buy in our stores are really studied, and we do not have to fear them. So another point is that GMOs are helping in everyday life. I have here an example as insulin, because the people who are suffering with diabetes mellitus are among us, and in Europe then they have enough insulin. But if we look into another nations in Africa and Asia, there are regions where's no insulin, and people suffering with diabetes are dying on everyday basis. And one of the solution is to make insulin producing bacteria, and to take this insulin in these areas and helping these people. But we cannot produce insulin in man and somehow I don't isolate it or purified it. It's not possible. Another point: breeding is an evolution driven by humans. Yeah, there we are touching the theme or topic of breeding, because I'd like to compare the GMOs and breeding, because the functionality of these processes is near themselves. So I'd like to talk about breeding and how it's more connected to nature, because breeding is just selection, yeah, I will talk about this later. So, and the difference between GMO and breeding, that's the last point, and we will talk about it on this slide. So our program is divided into two parts. One part is theoretical. We talk about GMO, we talk about breeding, we talk about transcription and translation, we talk about CRISPR and Cas, we talk about GMO and breeding. And now I'd like you to open Slido and ask the first question. And why do you think is the mRNA so important in the connection to GMO, or in the connection to transcription, translation itself? And that's the question I'm asking the students in the program. So the question is, why is the mRNA so important in the expression of genetic information? What do you think? Or you can answer if you want to answer with your hand, because I expected that there will be more people. Yeah, that's the first answer. Thank you. Yeah, I maybe put the question wrong, or alternative splicing. Yeah. The alternative splicing is a very good point, because if you want to produce insulin in bacteria, there is the little problem that you have to make there is no spliceosome, so you have to splice the DNA before and then put it into the bacteria, or the bacteria won't be able to produce the insulin. Yeah. That's cool. Yeah, thank you for the answers. The thing I was aiming to is that I'm sorry, where are we? Good. So I'm sorry. So yeah, mRNA is so important, but the thing I was aiming for is that we do have DNA in the cell nucleus, and it's here stable, unchanged, and there's the space that's the DNA like in their home, but we have the translational apparatus in the cell, so we have to take the information from the nucleus and put it out. So we have to transcribe it into mRNA, and so it could go outside the nucleus. So that was I was aiming for, but that's my mistake. And there's another question: where do I interfere in the process of expression of genetic information? Can I interfere in mRNA? Can I interfere in protein or DNA? What do you think? What do you think is the best space way where I can put my fingers into the expression of genetic information and change the protein itself, or change the trait, or change the cell, or make genetically modified organism? Yeah, we don't have much time, so you want to use the Slido. So yeah, the point is, and that's for children. So I'm just showing you the questions we are trying to answer in the program. So the best way to interfere in the organisms is the DNA, because that we can. It's the most stable way to modify the organisms. Yeah, and so these are the topics we are talking about with students at our program. So yeah, this is well, yeah, I have it here because this is the part of the video Eliška was talking about. We have at the start with DNA, then mRNA with some head, which is information that it has to go outside the nucleus, and then there is a full protein. Yeah, and then I ask children, what do you think can happen when you change one bit of the DNA, one amino acid in the hemoglobin? What could happen? Yeah, and the answer is that we will be not able to transfer oxygen. And the thing I'm aiming to is sickle cell anemia, because actually the hemoglobin it's altered in this disease, and not only the protein has another shape, but also the red blood cells are like sickle shaped. So that's the thing we have here. Yeah, and then as a practical part, Eliška talked about the GMO game we have, and you're invited to the exposition at 2 o'clock and talk about this with Eliška. So, and then we have programs for primary school. The first is about the cell and about the basics of genetics, or about the molecular genetics more than the Mendelian. In this program, we use the model of cell, which is this, as Eliška was talking about, the city, which is actually a cell and comparison of compartments within a cell with buildings inside of the city. And the second program is about the bees and about meteorology. I am beekeeper myself, so we are talking with children about beekeeping and more about the bees and how they live, how they are interesting, and the socialism in this organism is quite astonishing. So that's what we're talking about. Yeah, and now it's time for some discussion. If you have any questions, or not to me, or it could be question for girls.
Yeah, I think we prepared great discussion, just so you know. But we can do it over lunch, because that's not that very many of us. So we will have lunch right now, and we will be ready, like the three of us, in the exhibition, starting at like 2 o'clock. And before that, during lunch, we have some questions. Maybe we can just show the questions so that you are inspired to talk to each other and to discuss these questions.
Yeah. That's the thing is, I'm sorry to steal your word, but this is a question I'm talking about with students in the program. So it's from the program. So, and you can talk about it.
Yeah. So if you visit a local store to buy tomatoes and they have normal and GMO ones, what type do you choose? Maybe we'll just put all the questions on one slide afterwards, so that it can stay here, and when you're somewhere having lunch, you can just take a peek and say, yeah, this question I wanted to ask. So I'll just tell you briefly now. Second one is if we should discuss controversial topics in biology class. If biology classes are the place to do this, because at least in Czech Republic, I don't think it's not done very often. And some parents might be actually mad about bringing these topics, and they're controversial, so I understand. So like if we should discuss those, what controversial topics in biology would you like to be taught in schools? Like, we decided for GMO because it's very current topic, I would say. But what other topics you think of that you think would be good to be taught? And the last one, and for me the best question: who or what ignited your passion for science? if you could think of that maybe for yourself, maybe share it with others here during lunch. For example, I had a very good teacher at high school who basically he made me apply for molecular biology, and I'm still forever grateful to him. And he was very passionate about what he was doing. And and second thing, I was given a perfect encyclopedia when I was 6 years old, and I was learning to read from an encyclopedia from my grandmother, and no one understood why would she give me encyclopedia to learn reading, but I did that, and it's why I'm a scientist now. That's what I think, at least. So who or what ignited your passion for science? Yeah, and that's all from us. And we can head to lunch. Or Blanka, do you want to? Yeah? Okay. So thank you for your attention.